Imagine a tiny organ with the power to shape human history and spark fierce debates. The penis. Close your eyes for a moment and picture an ancient Greek statue. Notice how sculptors captured every curve and vein, and how those shapes spoke of power. Fertility, even the gods themselves. Now fast forward to a modern lab, where a PhD student peers through a microscope at a cross-section of penile tissue, marveling at layers of smooth muscle, blood vessels, and a network of nerves so precise you could trace them one by one. Our story starts in the dusty archives of 18th century explorers, who collected penis specimens from animals around the globe. They wrote letters that read like treasure maps, descriptions of walrus, elephant, even giraffe penises, each more bizarre than the last. These early naturalists believed that if they could understand the strangest shapes, they might unlock secrets of human reproduction. Today, we know those shapes evolved under sexual selection, a dance of biology and behavior that pushes organs to new extremes. Now imagine you are that curious explorer. You stand at the edge of a wet lab. On the bench sits a set of 3D printed models of human and primate penises. Your task: measure elasticity, compare glans geometry, record blood flow patterns under different pressure. You attach ultrasound probes
on a screen.
The cavernous spaces light up, expanding like tiny bladders and contracting as you remove the stimulus. You realize every pulse, every squeeze is a miracle of biomechanics and neurochemistry working in harmony. When we talk about the penis, we often stick to anatomy textbooks or awkward joke, but as researchers, we chase deeper questions. How do stem cells repair smooth muscle injuries in the corpus cavernosum? Could we one day regenerate functional erectile tissue for patients who lose it to disease? Which ion channels control the swift changes in blood flow that turn a flaccid organ into an erect one? And what can comparative studies tell us about human sexual health, about conditions like erectile dysfunction or Peyronie's disease? Your next experiment could link genetics and physiology, map gene expression in penile tissue, and then test how tiny chemical signals trigger nerve-muscle connection. You could collaborate with engineers to build microsensors that record pressure inside real-time sample. Or perhaps you dive into anthropology, tracing how cultural attitudes shaped medical treatments across centuries. Take a moment and feel that spark of wonder. Behind every pubescent or aging penis lies a world of cells, signals, and stories waiting to be explored. Your challenge is to bring together evolutionary theory, cutting-edge technology, and compassionate care. Push boundaries, ask bold questions, and remember, this humble organ still holds mysteries that could redefine our understanding of human sexuality.